A lot of what we do is early detection monitoring, and essentially what that is we go out, we do surveys trying to find the next potential invasive species or identify any potential range expansion of ones that were already here. And the reason why we do that is really down to this invasion curve. It's probably the most famous figure when it comes to invasive species. And the whole point for what we do is we want to catch something at this point here. When you catch an invasive species early, it's easy to get rid of it or manage it at a lower cost. But the longer it takes, the cost goes up, and also any potential impacts that species could have also get more severe, to the point where you see things like zebra mussels around gobies that we can't get rid of anymore, and it becomes a local management that's very costly. So like I said, the whole idea is that we want to improve our control chances and also limit any potential impacts a species could have by getting here. And one of the tools that my office uses to do so is larval fish surveys. So ichthyoplankton are larval fish. They're just the little fish that we see every spring. The surveys themselves can be very insightful, especially when you're looking into fish abundance or year class strength. But for us, the reason why they're so useful is that fish tend to be the most abundant in their larval stage. And an invasive species is really looking for a needle in a haystack. And invasive, at least for larval fish, because they're so abundant, it gives us that opportunity to kind of look through as much hay as we can to find that one needle. And so the year I'm going to be focusing on is our 2017 sampling season, where we sampled in both Lake Erie and Presque Isle Bay, the Buffalo River and the Upper Niagara River, and also Lake Ontario, where we sampled in the Genesee River, Irondequoit Bay, and the Oswego Harbor. We used 4 gear types in this sampling season: push nets, which are these nets right here, along with larval seines, bongo nets, and light traps. So we did work at night and during the day as well. And for identification, we did a morphological technique that we did in-house at our own lab in our office, and then also a genetic component that we sent out to the Northeast Fisheries Center in Lamar, Pennsylvania. All right, so the first technique I want to talk about is the morphological, because it is what we did first. We would collect the samples, and then we would bring them back to our office, where we would pick through any vegetation that we collected, or zooplankton as well. And then from there we would ID down to family, just because it was easier to separate everything out, and then from family to species. Like I said, everything was done in-house, and we used a setup very similar to this one here, where we would have a scope that had a camera attached to it that would project the picture onto a computer screen, so that we could take pictures when needed, measure a little easier, and also see some of those really fine details just a little bit better. So with every technique that I'm going to talk about, I also want to get into the advantages and disadvantages of them. And for morphological, some of the advantages are there are key characteristics that can really distinguish the families in the Great Lakes from one another. And for us, that's really helpful because it just makes the whole process a little bit easier. And for those techniques, for those characteristics, the ones that I relied on the most were preanal length, and then also myomere counts, both preanal and postanal myomere counts. And what we saw for this is that even if you had one of those characteristics overlap for a family, the other 2 wouldn't. So you could really have some confidence that if you found a family, that you could believe that's what it really was. Another one is the keys that are available. Our et al. Is probably the most famous for Great Lakes larval fish identification. It does a really good job for native species in the Great Lakes and really hitting those characteristics that can separate species out. But if you do ever run into any issues, there are other offices who have published online pictures of different larval fish. So if you have a question, you can always kind of fall back on those as well. And then also, it's just the most cost effective. This is all you really need. You need a person to do it, a key, and a microscope. So if you're worried about budget in your project, this is the most cost-effective way. Unfortunately, there are disadvantages as well, and probably one of the biggest ones is that larval fish just grow really quickly, and they don't really adhere to a key. You know, we have a good set of guidelines that we can use, but fish grow at different rates. And sometimes you'll see a fish that doesn't really match up with the description that's given in the key, and you kind of have to use some other resources to figure out what it is. Time of identification is also a big one. So in 2017, I had roughly 9,000 fish that I needed to identify, and unfortunately, they're not all in great condition. You'll get fish that are somehow damaged, they're missing a fin, or they've been in alcohol too long, so their pigmentation is starting to disappear. And it can just make it a very time-consumptive process, especially when you could have a fish that's in a family that has several species, and you're trying to figure out what your one damaged fish is, knowing you have, 9,000 behind that one that you have to get to too. And then also for my program, the keys, they're great for native fish in the Great Lakes, but that's not what I'm doing. I'm looking for the next invasive species in the Great Lakes. And so that can be a little tricky because there's not really a whole lot available to me to be able to do that. And then also sometimes the descriptions in the keys can be a little difficult. What I mean by that is if you look at brook silverside, in the Great Lakes, or at least the areas that we sample, it's the only one in its family. And the description in the key, along with the sketches that they give, actually match pretty well for the fish itself. You can see the body shape is relatively the same, the placing of the fins, relatively the same. But then you get over to things like cyprinids, where there's a lot of different cyprinids in the Great Lakes. And unfortunately, you can get things that have some damage, some bent tails, and you try your best to get down to what it is, but there's a lot of overlap, especially in this family, in the keys for some of those key characteristics. And also all the pictures and sketches they have look about this. So you have to fall back on that. It's not much to fall back on. So the next component that we did was a genetic component. Like I said, it was sent to the Northeast Fisheries Center in Lamar, and it was done, we would ID all of our fish, and then we sent a subsample of those fish to them so that we could compare what I was saying to what genetics was saying. And what they did is they used the Cohen gene for the genetic identification. And how this is done is they would amplify the standard Co1 barcode region using universal fish primers, and then use the sequence data that they collected to then align that against a reference sequence database, which was the Barcode of Life data system. And all of the genetic barcodes in that database are all obtained from adult fish that were positively ID'd, mainly because adult fish are easier to ID than larval fish, so you can have more confidence in your barcode if you're getting it from an adult. So advantages for this, like I mentioned, that the BOLD data system, it's 20 years old, so it's been around for quite some time. A lot of people do use it. And also, probably one of the biggest ones is it had a quicker turnaround time for us than it took for me to identify. We started picking in July, and I wasn't done with the identification until March. So if you're worried about time, this can be a little bit quicker than morphological. And then another big one is that BOLD data system is nearly complete for fish. So if you're getting a species, you can have some high confidence that that's really what it is. Going along with that, for the disadvantages, you do have, especially for cyprinids, a lot of closely related species, and that can cause some issues when it does come to genetics work as well, so just keep that in mind. Another big one for at least this technique is you can only do one fish at a time. If you have 9,000 fish, it's also not going to be super fast because it's just one fish at a time. Cost per sample, it can be pretty pricey, so if that's something that you're worried about, just keep that in mind as well. And then also you have the potential loss of the fish. So in our case with invasive species, we want to be able to document things as best as possible. And in order to get the DNA out of a fish, you have to grind it up. And if you had a question about something and the genetics don't bring anything back, you better hope you took some good pictures because that fish is now gone. Right, so like I said, in 2017, we did this comparison study to kind of see how our morphological ID lined up with the genetics ID. And what we saw is that at the family level, we had a 97% agreement rate, which is kind of what we were expecting, because like I mentioned, with those characteristics at the family level that really distinguish them, and along with the fact that the bold data system they used has a high confidence behind it, we were kind of expecting to see a pretty good agreement rate at family level. Fortunately, by the time we got to species, it was only at 40% agreement rate. And what we saw is that a lot of the disagreement came at those closely related species. So if I was calling something a pumpkin seed, genetics was saying it was a bluegill. And same with the shiners, there was a lot of different identifications going on between the two techniques, which after reading several other papers, we did notice that this is pretty common, and this is roughly what other papers get, about 40 to 50% agreement rate of species. So for future directions, if you're doing larval fish surveys, your project's really going to determine what technique you're going to want to use. So if you're looking at Great Lakes fish, ours is a great key for that. But in our case, we're looking for invasive species, and it would really be helpful if we could collect fish in, say, June, July, and have results back in September, and there is something new there. It just gives us the opportunity to go out and do a potential eDNA and adult surveys and kind of figure out where it is and what we can do to manage it. And so a technique that we've utilized in the past couple of years is qPCR, which is just another genetic identification technique, and it just uses a different genetic sequence than the CO1 gene. Again, it has its advantages and disadvantages. Probably the biggest one for us, because we collect so many larval fish, is that you can analyze a mixed sample, so you don't have to tease one fish out at a time. You can process a whole sample at the same time. This allows for high throughput and also a really quick turnaround time, which, like I mentioned, for invasive species is important for us. Some disadvantages is you can have some potential questions about having a representative sample. What I mean by that is, if you can see down here, there's one big fish in with probably 40 or 50 little fish. So that big fish is going to supply a lot more tissue to the sample itself, which can lead to some issues of it potentially overshadowing the genetics of the other species in there that are smaller. Another disadvantage is also some taxonomic resolution issues. If you look at this sample here, it had 725 fish in it. They may not all be the same species. There could be several species in here. And because there's so much tissue to work through, you could potentially have some misidentifications or some issues with that with taxonomy as well. And then another big one is for this genetic sequence, the data set is not as complete as the CO1 gene that we used for our 2017 sampling season. And so currently in my office, to kind of fill some of those holes that we have, we are collecting invasive species based off our top priority species list, and also species we're worried about for possible range expansion from other people around the country, but also from other countries too. So we're trying to fill that hole in this technique so that we can utilize it more effectively and also kind of increase our chances of finding the next invasive that we're concerned about early enough that we're able to do something about it. Oops, sorry. And then just quickly, I have a couple of acknowledgements. All of our work is for larval fish is funded by the Great Lakes Restoration Initiative. And then I want to thank everyone that helped us at the Northeast Fisheries Center, especially Erin Malloy, who was one of my co-authors, and Meredith Barton. They did a lot of work for us getting these samples processed. And then also just to all my coworkers who helped collect these fish, but also helped pick through the samples so that I didn't have to. All right, with that, I'll take any questions. We
got about 5 minutes for questions.
Yes.
I was wondering, did you find any potential invasives?
Nothing new. No, actually in 2017, I identified a tube nose goby from Presque Isle Bay. They're already there, so it's nothing new. But we hadn't caught any adult tube nose from there yet. So we got a larval fish, and then the following year we got an adult fish. So that's pretty cool.